Hi, I'm Archana Jalligampala, a postdoctoral associate in the Department of Ophthalmology and Visual Science at the University of Louisville. My research focus aims at using various gene therapy approaches for the treatment of inherited retinal diseases. Inherited retinal diseases are a diverse group of progressive, blinding genetic diseases that can manifest any time from birth to late middle age. In the United States, the inherited retinal diseases result in a socioeconomic burden of about $33 billion annually. More than 200 genes have been found to be mutated in these diseases. Socially, these patients find it very difficult to perform simple day-to-day tasks, and economically, they find it difficult to get employment, resulting in depression and low self-esteem. One of the most common inherited retinal disease is retinitis pigmentosa, or referred to as RP, which is characterized by the progressive loss of photoreceptor cells, primarily the rods, followed by cones, eventually leading to blindness. The prevalence rate of RP is approximately 1 in 4,000 people worldwide. The RP patients often report impaired night vision, loss of peripheral vision, also known as tunnel vision, and progressive deterioration of the central vision. Over 80 different genes are associated with RP. The autosomal dominant form of retinitis pigmentosa, also commonly referred to as adRP, accounts for about 30 to 40% of the RP patients, where only one copy of the mutated gene is enough to manifest the disease. Mutations in the rhodopsin gene, also known as Rho, are responsible for about 25% of the cases of adRP in the Caucasian. The P23H rhodopsin mutation, which is a proline to histidine substitution, is a prevalent form of adRP in North Americans. The P23H adRP patients carry one copy of the wild-type rhodopsin gene and one copy of the P23H mutant rhodopsin gene. The mutation results in mislocalization and misfolding of rhodopsin, resulting in rod degeneration and eventually blindness. Currently, there is no definitive cure for these patients. In collaboration with the National SWINE Research Resource Center, our lab has developed a large model of adRP that expresses the human P23H rhodopsin mutation. The transgenic pig has 6 copies of the human P23H rhodopsin mutation and 2 copies of the wild-type rhodopsin. Surprisingly, although there is a fair complement of rod photoreceptors at birth, there is no functional rod-related responses, and these further do not develop with age. These factors, along with the pig's eye size, make it an efficient large animal model to test the efficacy of various gene therapy approaches. So how does gene therapy work? In gene therapy, one can replace the disease-causing gene with a healthy gene. One can add a gene that can help the body fight or treat the disease, or one can turn off the disease-causing gene. To insert new genes directly into the cell, scientists use a vehicle called vectors, which is genetically engineered to deliver the gene of interest. Viruses have a natural ability to deliver genetic material to the cell and therefore can be used as vectors. Over the past decade, the field of gene therapy has seen an explosion of cutting-edge technologies such as gene editing. Gene editing uses various molecular tools to precisely target the problematic gene and create a cut or break in their DNA. The best-known gene editing systems are CRISPR-Cas9, meganucleases, and zinc finger nuclease. The meganucleases are DNA-cutting enzymes that can recognize and cleave unique large DNA targets of around 14 to 40 base pairs. The Rho12 meganuclease, a proprietary of Precision Bioscience, is an engineered meganuclease that is packaged in a self-complementary AAV5 virus and is driven by a promoter that targets the photoreceptor specifically. The Rho12 is delivered in the pig eye subretinally in the first postnatal week. The Rho12 recognizes and cuts the human P23H allele, resulting in a double-stranded DNA break. The cell's repair machinery inactivates the mutant P23H gene and causes degradation of the human P23H rhodopsin, resulting in the expression of the wild-type PIG rhodopsin. 1-year preclinical results show that the Rho12 not only rejuvenates the rod photoreceptor structure, but also its function, and this function is maintained up to 1 year. Such promising results suggest the level of gene editing achieved in this study can effectively treat rod degeneration and facilitate visual improvements in P23H adRP patients. Thus, my long-term research goal is to develop treatments for inherited retinal diseases in collaboration with industry and academia that can improve the quality of the visually impaired patients. Thank you for your attention.